Okay, so let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us the 76th seminar, and I call it a University of Michigan Day. I have been in Europe for 12 days to give 4 invited talks so far, and then now I will enjoy my weekend. And then I enjoy my also Easter vacation in Paris, and with one of my college friend, I would say the best friend, who happened to be working in Germany starting this year. I love trips because I can see nature, history, diverse cultures, and most importantly, people. Since the summer 2021, I have been traveling to meet people, share my vision and work. Last year, I was out of town for more than 160 days, and I will be out of town again for more than, I guess, 140 days this year. I just want to regain human connections, which we missed during the pandemic. So let's reconnect and meet in person somewhere in conference or seminar and anywhere. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Professor Pete Tessier. I mean, he doesn't need any introduction because we all know him. He is an Albert Mattox Professor of Pharmaceutical Science and Chemical Engineering at University of Michigan. More importantly, he mentioned he likes to be the tallest person in this Zoom room, although we cannot compare. I know him very well, and I am saying he is a tall guy, but I remember him as a smart and kind person with very nice voice. So Pete, I truly thank you for your time and for your contribution to the scientific society and community, and the virtual podium is all yours. Thank you.
Well, thank you very much. And I am a tall guy. I have to say, on Zoom, I've had some funny experiences. We've lived through COVID over the last 3 years, and I met a lot of people that thought I was a short guy because I sit behind a desk and my head's often cut off here. So I'm joking. But anyway, it's great to be virtually together here. And I agree with the sentiment of I hope everybody is well and engaging in person as much as possible. So it's my pleasure to be here and give a short talk in the beginning of a really true emerging star and a personal friend of mine. And so it's really particularly special to be here today. I wanted to share with you a few thoughts that I had about a scientific career. And there are a few observations I've made in my time that I hope will be helpful to you. So during my journey, I found a few things that perhaps will be helpful in thinking about your journey as a scientist. Now, I'm going to have time to cover sort of the first 3 of these, things about creativity, growth, and discovery. I won't have time to go through the last 2. If anybody's interested, feel free to reach out, and I'm happy to discuss. First one I wanted to talk about is creativity. And there's an interesting principle here that creativity is actually contagious, and it's actually hard to define, but when you're around highly creative people, it has an effect that you can't really explain, but it's quite useful. So I started graduate school in 1998, and I had the good fortune to work with this person. He was not my advisor. He was a research scientist in a lab that I was working in. His name is Orlin Veliv. Probably some of us might know him. He's at NC State right now. Pound for pound, one of the most creative people around. And so when I started doing research, I was working on a specific problem related to protein thermodynamics. We were trying to measure protein self-association and developing methods for measuring these types of interactions. It was interest to try to crystallize proteins by being able to measure these weak self-interactions. But on the side, I started working with Orlin. And that's where I started to be around this highly creative person. The guy just was always had interesting science going on. And actually, I had the good fortune. My first paper was in Nature with him on a kind of material science story. I don't have time to go into it today, but just being around a person that thought that creatively about science made me want to think like that. And it just, you start thinking differently. And again, I got infected with scientific creativity. So for my research, what we were trying to do is measure protein interactions to try to promote and predict conditions that would lead to protein crystallization. But there was like a problem in this field. And the problem was that all the methods that people were developing, including what I was developing, was very slow and laborious and had very low throughput. So because I was working with Orlin and he had infected me with his creativity, I wanted to think like he did, and actually led to an interesting story where we developed these nanoparticle conjugates where we could conjugate proteins to nanoparticles. And we got this very weird behavior that exactly at conditions that would lead to crystallization, essentially that we're in this weakly attractive region of the second Vierer coefficient. It's beyond sort of the details today. We got a very unique self-assembly behavior where these particles would associate and tell us essentially where the protein wanted to crystallize. And that led to, subsequently, we developed a number of assays that were related to that with different pharmaceutical companies, again, using these nanoparticle conjugates. And that led to some interesting outcomes. So we solved the problem, and the problem was we could use ultra dilute antibody concentrations to get at these self interactions that are important for formulation. And based on this, actually, this assay, one of the assays we developed, ended up in a high-profile paper and is sort of broadly used for identifying drug-like antibodies. So our assay was one of the assays that could identify antibodies that are used in people that are sort of have superior biophysical properties. And in fact, this assay has been used by Pfizer, surprisingly, to predict pharmacokinetics in mice and even in people. So essentially, antibodies that have sort of favorable pharmacokinetics, you could identify using our assay. The point is not about the details. The point is that the research that came out of this comes back to what I'm going to call the creativity proximity effect, that being around highly creative people makes you more creative. It infects you. It makes you think of things. And that's sort of just, this line of research is just an example. Never would have done any of it had I not been around this highly creative person that made me want to think creatively. The second thing I want to talk about is growth. So growth usually comes from being outside of your comfort zone. If you work in the same area a lot, you could become an expert in that area, but it's very hard to go beyond. So what I want to encourage you is think about seizing opportunities that are different than what you already know. So when I went to do a postdoc, I was a chemical engineer. I knew very little about biology, biochemistry. I worked with this lady, Susan Lindquist, and talk about moving out of my comfort zone. Wow. We worked on a specific problem related to prion proteins and some physical chemistry related to prion proteins. The thing was, when I went and did this, I essentially knew nothing about all of the things I needed. So on a percentage basis, I knew essentially nothing about all of the techniques that were the basis of my postdoc experience. And in fact, kind of a funny thing happened. So I applied for a postdoc fellowship, an NIH postdoc fellowship, and this lab was huge and had lots of people way smarter than me. But I got the highest score ever on this for that lab on my fellowship application. And the reason was, if you read the reviews, it's that the reviewers would say, wow, this guy doesn't know anything. And the training potential here is extremely high. So we're going to score him very high. So even the reviewers said, wow, this guy's way outside of his comfort zone and certainly way outside of his area of expertise. My postdoc was very interesting. It led to a Nature paper on our research. And this really, the culmination here, and the point I want to just make is that moving outside of your comfort zone is very helpful to develop you as a scientist. I call this the expanded horizons effect. And the last thing I want to tell you about is discovery. And discovery, really in science, is strongly improved or aided by being around people that think differently than you. And just one experience I had was working with this guy here. This was from a newspaper article about how he can solve a Rubik's Cube in 2 minutes. I can tell you, I've seen him do it much faster than that. It's limited by how smooth the pieces move on the Rubik's Cube. So what happened was we were working on directed evolution of antibodies, and we had this kind of observation that if we started with an antibody and introduced mutations and made large libraries and then did selections for binding to our target of interest, we always got this effect that whenever we find a variant that would be better or affinity matured, it would also be less stable. So there's this very strong affinity stability trade-off we were seeing, and we just couldn't understand it. I don't know how else to tell you, as we just were totally lost in what was going on. And then we had a meeting with somebody who just thought completely different than we did, that didn't bring any of the biases that we had, that didn't think, weren't deep into the problem like we were. And afterward, he helped us understand this principle, which led to a lot of interesting science. And the principle is that a starting antibody has a well-folded structure, but it doesn't really bind the antigen, or binds very weakly. But as you affinity mature and improve sort of the binding to this target molecule, the structure of those loops often is not ideal anymore for the underlying scaffold. And it's important both in nature and in the lab that one accumulates these additional mutations that reset the scaffold back to this folded state. And once he explained that to us, like he had a hypothesis, the light came on and led to lots of interesting science. The point is, we could not have got there had we not been around people that just think completely differently than we did. So I call this the diverse feedback effect. And don't miss how significant this can be in your scientific journey. So because of time, I have to skip the last two. I just want to summarize here that in 4, I would just say scientific impact, the impact of your work is often going to be influenced by looking for opportunities that arise that you don't expect to arise. So being able and willing to rapidly adapt and not just stuck only on the one thing you're kind of thinking is very important. So that's something that I found to be very helpful. And then finally, some advances that you will make in your career will only come if you just stick with it. So there's going to be some problems are important, but you do not know how to solve at the moment. And unless you stick with it over a longer period of time, it just doesn't happen. And so for us, that has also been something that's been important, that for some things, we've just stuck with it for a long time. And only 10 years later, we finally able to solve that problem. So that's something else I'd challenge you to consider. So finally, I just want to thank the team of students in the lab and my alumni. And again, it's an honor to be here, and I really feel privileged to go before this outstanding scientist we're going to hear from now. Thank you.
Oh my goodness! You know what? I would say this is one of the best talks in recent, month. I mean, absolutely agree with you. I feel like I'm getting younger every day whenever I talk to creative young people, and their crazy ideas, sometimes not feasible, but still that feed me
Right.
new idea, new direction. Also, I also believe you mentioned feedback, but I would say, I have the great idea whenever I do random chat over the beer or over the coffee,
Exactly.
rather than constantly
Right.
talking about something with someone very closely related to my research.
Right.
So that's why I also love traveling, because I meet so many people with so many different perspectives, getting so many feedback. And absolutely, I love your summary. That is well thought, well designed, and I believe that is lesson to all the young people. Thank you so much. That is
Sure.
absolutely wonderful. Thank you.
Thank you.
Okay. So now the main speaker of today with a longer introduction, Dr. Ratul Chowdhury did his PhD with Costas Maranas and Penn State University, chemical engineering, where he developed several integer optimization-based pipelines to design proteins. So another protein that is important, common, interest between these two speakers. From 2020 to 22, he was a postdoctoral research fellow at Harvard Medical School in the lab of Peter Sorger in the Department of Systems Biology. There he developed an AI-guided protein structure prediction tool from single sequence only. That's very, very challenging work, but he did it. He was also developing computational medicinal chemistry pipelines for non-opioid drug discovery. Currently, he is an assistant professor, just brand new, and congrats again, just less than 1 year, at the Department of Chemical and Biological Engineering at Iowa State University, where he is developing machine learning pipelines for protein design, where the focus is to decide if machine learning is at all required for designing certain classes of proteins. To me, AI will be the next future thing for the protein design and other. And he is one of the truly rising star. And I'm so excited to listen to his talk today. Ratul, thanks for your time today, and please take it away. It's all yours now. Thank you.
Oh, thank you so much. That was a great introduction. Let me see. Let me try to share the screen and this. Can you see my screen?
Yeah, wonderful. Go ahead.
Okay. Yeah. So thank you for giving me the chance. I will try to talk about some of the things we're trying to develop here, some of the things we already know, and try to weave a story out of what we can do. So the title of the talk is Single Sequence Protein Structure Prediction, but we'll weave a story as to how I got to this. Like, this was not the first thing that I started working with. So currently I am at the Iowa State University. I'm a part of 3 centers, which is inside Iowa State: the Nano Vaccine, the Institute, the CBERC, the Bioeconomy Institute, and TRAC, the Translation AI Center. So in terms of work, so I just wanted to first show, okay, so proteins are, so what are proteins? I think majority of the crowd knows what the proteins are, but still, proteins are this unidirectional polymers where they are made up of, like, they look like a beaded structure, like a beaded necklace, where they fold and refold upon itself to get a specific structure. The structure is pretty important because structure determines function and how they can shape shift. They don't necessarily stay as stringy polymers. They become these precise shapes. And the moment you make a small change somewhere here or somewhere here, that can cascade down the protein backbone, pretty much like the example that Pete was talking about. So when you make a change, even with the vicinity of the protein, it can undergo conformational change. There's the same thing that we see multiple times. This is the popular protein now. So this is for the spike protein for SARS-CoV-2. So these are 3 identical sequence proteins. These are trimers. In the presence of each other, one of them, they actually wrap around each other, but when left alone, they would not be doing this motion. So proteins undergo motion, and understanding structure gives us an insight to function. So the story that I'm going to talk about is that, so we'll look at different classes of proteins. So first we'll look at proteins where we make little changes to existing proteins to make sure that the structure is intact, and then endow them with small local changed properties because we've made little changes on them. And then the next story would be where we make a lot of changes on an existing protein, make them do completely different things that they were doing originally in the living organism. And then the third part would be to sort of, motivate what we learn from these kind of examples, where we drastically change proteins and make them do things, and make non-protein molecules like synthetic architectures, which behave like proteins now. And then on the right-hand side, this part of the journey is where I typically show some data-intensive techniques, as Tessier rightly said. So machine learning, AI, all of these is becoming important. I will try to sort of throw some light as to, how one needs to carefully tread in terms of using these tools rather than taking them at face value. You need to be a biologist first, then a machine learner to be using biology, computation in biology. So first story, so we make small changes on existing proteins. So these protein classes are enzymes. So we worked with this to develop this tool called iPro plus minus. So enzyme engineering as a domain has been like quite popular. We make enzymes catalyze different reactions that they originally do. To make do that, we do small number of mutations on them. But here, so there were a lot of tools that are present for doing protein design. But we were saying that when you look at classic protein sequences, let's say this is a protein sequence from humans, and these are the same protein sequences from different related organisms. This family sequence alignment tells you that it's not just the alphabets that change, the length also changes. So the starting sequence, you can see there are dashes, which means this is shorter than other sequences in the family. And then there are dashes over here, which means this is longer than, the other members of the family. So that sort of tells us that if we leave out the possibility of making insertions and deletions, we lose out on 62%, as much as 62% of the whole family members, because you're just stuck to this family length, which is 275. This is for an example case of TEM beta lactamase. So we designed this pipeline which can accommodate insertions and deletions of variable lengths. So first question is, how do we decide where to make this insertion and deletion? So this could go like, one could be very, picky about it and go with a detailed route. But as a start, we made it that you can indicate from a multiple sequence alignment, what are the positions where you can make an insertion or a deletion. So now the next question is, to make an insertion and deletion is slightly more subtle task than a substitution, because when you do a substitution, you're just essentially changing the side chain, fixing the clashes, making sure the structure prediction is correct. But in case of an insertion and deletion, you actually have to break open the polypeptide chain, add a new amino acid, or remove an existing amino acid, then join it back. So in order to do that, we sought some sort of inspiration from a robotic arm algorithm, which is an inverse kinematic problem, where there are flexible elbows which are reaching a fixed target by minimizing the energy that's consumed. So we thought that the two snipped ends of the enzyme where the insertion or deletion would happen, one would serve as the target, the other would be the movable arm, and this would happen iteratively and alternatively. So in a visual depiction, this would look like this. We induce a break where we want to make the insertion or deletion. And then after making this insertion or deletion, we would try to join the ends by performing iterative movements of this flexible arms. While doing this, we always make sure that we are not violating dihedral angles as dictated by the Ramachandran plot. So essentially, the brief overview is, when we are making an insertion, we were just doing a glycine insert, because the glycine can then be replaced by different amino acids if this insertion is tolerated for the enzyme to become improved for the task it's at hand. So, and then there is deletion task is much more simpler. You delete the amino acid of interest, and then you join the flanking backbones. So there was an existing tool called iPro, which was iterative protein redesign and optimization from our lab. So now it had this extended capability to perform an insertion and make that insertion become something else, and also alternatively do a deletion. So we tested this with again, the family of TEM beta lactamases, for which a lot of mutant data was available and functional assays were developed and reported. So we wanted to check the binding efficiency with this inhibitor molecule. And in order to do that as the fitness, what are the insertions and deletions nature has already seen during the course of evolution. So we are seeing that when we start the algorithm with the wild type, it picks up 5 out of 7 naturally occurring insertions and 7 out of 9 naturally occurring deletions, along with other substitutions. So And the second case where we again validated this was to explain the substrate specificity switch. There is this 4-coumarate-CoA ligase enzyme, which originally binds to the smaller substrate, 4-coumarate. We wanted it to bind to synapate because this has some lignin valorization outcomes. So, okay, we want this enzyme to work on this larger substrate. There was some literature evidence that these two positions might be able to tolerate a deletion with the little sequence alignment that we were able to get. So there was one paper which actually showed that that there was when the synapate binds, it happens at the loss of coumarate-CoA activity. So we did not have a experiment like a structural model to explain that. So we were able to simulate our algorithm, and we've identified that this particular mutant V345, this particular deletion, when this valine is deleted, it opens up much more of the pocket volume, thereby allowing a clash-free stabilization of the larger substrate. That's why this deletion is preferred and also justified. So these were two spot checks that we did with our algorithm at that point. And then we've been using this at a broader scale in collaboration with Dr. Brian Flegger across different studies. So then coming to the next story, where we take a protein, where we make a lot of changes to this protein to give it a new property. So here we're shifting away from enzymes or biocatalysts to look at pore proteins which have a central pore in them through which things can pass. Okay, so this was in collaboration with University of Texas and UIUC. So the idea is that, can we take these channel proteins out from the context of naturally occurring membranes and use them as filtration units? So a wild type protein typically has like a bigger pore. We want to be able to design the pore interior, such that we can get a narrower pore and a hydrophobic pore. So a narrow pore is understandable, a hydrophobic pore primarily because the water is anyway coming into the pore because of an osmotic gradient. So once it's in, you don't want it to stick it longer. You want it to go as fast as possible. So that's why the hydrophobic pore. So there's already a water conducting pore that's already well and popular. It's aquaporin. Its precise internal pore geometry permits only one water molecule to go through. You see this representative water molecule in yellow. So why not just use this if you want a precise filtration unit out of this? So actually, NASA was interested in using aquaporin for filtration of urine in International Space Stations to separate out sulfur and phosphorus to get to drinkable quality water. But the major problem was structural stability and the very hydrophilic pore wall. Structural stability is because these are like telephone cords, like old style telephone cords. If you pull them during sheer stress of operation, their pore sizes would deviate even by an angstrom. And that would lead to inaccurate filtration operations. So we said that how about we marry the two important properties? So we take a structurally stable scaffold, which is the outer membrane porine, which is a beta viral protein, and have built the internal selective pore geometry of aquaporin inside it. So and our collaborators at UT Austin said that if you make these pores, we can lay them out as flat sheet membranes on a porous support. We can scale this up. So okay, so the hypothesis was that we have this pore constriction. We want to jam this pore constriction as much as possible with hydrophobic residues to make the smallest pore. So the best case scenario was to jam it up with tryptophans. But then soon it turned out to be an optimizational problem, because when you jam it up with tryptophans, the consecutive tryptophans start clashing with each other, and some of them are pointing outward from the screen of the that you're looking at, and some of them start pointing inward into the screen, and the pore size is not what you expect. So soon it became an optimization problem as to how to place long and short amino acids, which are hydrophobic, in a pattern such that all of them contribute to the maximum pore closure. That's exactly what Pore Designer does. So we do this optimal placement of different amino acids at the pore constriction to control the pore size. And we were able to make the pore constriction as hydrophobic. This is experimental data, as hydrophobic as carbon nanotubes. And we also have experimental data, which I'm not showing here, to show the rejection. Like the transport properties were so good that we were able to separate out a 558 kilodalton, the smallest NaCl also. Well, this does not lay foundations for desalination, because if you desalinate water, like convert salt water into drinkable quality water using this, you'll be buying a gallon of water for $40 from the market. No one's going to do that. But what this does is set up the stage for us to believe that we can tune pore sizes reliably and efficiently. So we soon started collaborating with Imperial College London, where we are trying to look at designing pores for endowing pores which do not have DNA sequencing properties, to endow them with DNA sequencing properties. So to do that, so DNA sequencing happens in a way the motor protein sits on top of a pore, pushes down the nucleotide, and based on the nucleotide and buffer interaction at the constriction, it records unique current. So Oxford Nanopore is already doing this. This is no strange thing for anyone. They are doing with alpha hemolysin pores and MSPA pores currently, but there's a lot of patent and art around it. So we wanted to see that, okay, can we take a pore which does not originally have selective ATGC sensing mechanism and endow it with that property? So some of our initial results show that, like, this is a nonomer. So one mutation you make gets radially distributed across 9 monomers that guard the pore. So there were 3 mutations that we were able to identify, which sort of shows that, this is the green signal over here is from the wild type pore, where there is no discernible signal from AT and G and C. But these mutants show promise. We are yet to look into this in more detail, but this is promising. So then this is the third piece of the story that I said, like, now we understand somewhat as to how pores behave and how pore proteins can be tuned and modulated. But if you're talking about real filtration and separation and care about scale up, one question is, protein pores offer very limited usable cross-sectional area, because the whole of this purple or pink thing, as you might perceive, is just the protein bulk. It is not the pore. So this is all impervious. This is the usable pore. So how about we can make a synthetic pore which just occupies this much real estate? That way you can jam in more number of pores per unit area of the membrane material. So people have been looking at supramolecular channels, carbon nanotube porins, etc. So we said that, okay, we care about smallest pore, which just rejects water. We make a pore which basically just allows one water to go through, and that's it. And that should be the perfect design. So we did it, and it did not work. It did not work because you will see, if you look closely over here, the moment we synthesized this, they started agglomerating on each other. They started forming aggregates. And these aggregates had some sort of a collapsing nature on each other that reduced the pore size, even so little, by 0.3 angstroms. And that was enough to kill the water from going through. There is some energy minimization that happens here that forces them to collapse on each other. However, serendipitously, what happened was that this opened up lateral windows for the water to intercalate and go through. So that formed what you see over here, a lot of like a gyroid interconnected network of single file water transport geometries. So we then studied the biophysics and the chemistry and whatever is controlling the energy of activation of the single file water transport, and assessed the ion rejection through these materials. So we performed full AFM studies to show that these channels do aggregate with each other, and the moment the aggregation happens, the channel density is 500. That's when you see single file water transport happening. Before that, the permeation is actually less before aggregation happens. And this also shows that through these channels, the energy of activation barrier for the water to go through is much lesser than through the crevices or the defects of the lipids itself. All right. Okay. So we published this study soon. And then basically I moved on to studying how we can use data intensive techniques, how we can understand already published data, already reported sequence information, structure information, and biochemistry about proteins to design better and newer proteins. So, I mean, AlphaFold has definitely, taken the world by a storm, as it should, because it shows how you can convert sequence information to get very reliable structures, at least for homologous proteins. So like a small snippet of what's going on inside is that you take a protein sequence, generates its sequence alignment, take statistics from what occurs and what co-occurs every time, and use those statistics to impute some information about the distances between amino acids and angles between different amino acids. So how this essentially works is, let's say there are two domains which are separated by each other. Like this is a long sequence, this is the N terminus, this is the C terminus. But when you look at the structure, they actually are folded such that these two sections come close to each other. So which basically means for this structure of the similar family of proteins to be alike, all these interactions have to be retained. So if these interactions have to be retained, every time this R changes to something else, this also has to change to something such that they can still attract each other. So that's basically what AlphaFold is leveraging. So it is finding out the probabilities of co-occurrence of different amino acids across the polypeptide chain, and it's fitting a Gaussian. So the Gaussian sort of helps mathematically to convey the algorithm that if R and E are coming close to each other, their adjacent residues such as P and A would essentially have to be close to M and G. That's pretty much what it tells, and it helps the algorithm to learn faster. So now I think I've been able to convey that, how you can take sequence information to some distance-based information. And we have this basic functions which tells us, your van der Waals, your electrostatics and solvation, you can get some estimate about the energy profile of these molecules. And you can always perform gradient descent to refine the structures to get like energy minimized structures. I guess this is clear for now that, more the sequences, better the landscape description. You need more amount of members in the family of this protein to get a much more detailed description. So you get an ensemble of predicted structures, you refine, get the lowest energy conformation, that's it. However, this is the figure 5a from the AlphaFold paper, which basically tells that the moment you have very less number of sequences which are related to your target sequence that you're trying to predict, as this goes below a certain number, the prediction does take a bit of nosedive because it does not know where to get the statistics from. And also searching these databases is also a very GPU intensive as well as time intensive activity. So we said that, okay, while we have more and more sequence information to be leveraged using more and more compute, we can definitely drive towards accuracy for sure. But at the end of the day, the real biological process of protein folding happens at the level of a single sequence. When the protein is coming out from the back of the ribosome, it does not have information about how other proteins are coming out from the back of the ribosomes of other related organisms. All the information is in the single sequence and its surrounding pH and temperature, etc. Okay, so how about we push a method which learns only from single sequences and see how accurate can we get? That was the original objective. So we released this method last year. And then this was featured as the cover of Nature Biotech. So this is a single sequence method. Feel free to read about it and let me know if you have, if you find this interesting or have any questions on this. So what we are doing is that we are saying that proteins are not necessarily folding on an energy landscape. Energy landscapes are lossy. This is an information landscape. So if you are able to encode all the information coming from a single sequence, whether it's the information from the sequence, information about the biochemistry, information about bonded distances, you feed all of that there, and that way you can actually make a high fidelity reconstruction every time. So for the structure part, we were saying that, okay, how do we trace the backbone? So a protein structure, if you think closely, it is a continuous curve. So it's a one-dimensional curve in three-dimensional space. So, and these are spaced by different consecutive amino acids, and they are spaced by 3.8 angstroms every time, because the distance between C alpha to C alpha for every protein, for every amino acid is 3.8 angstrom. So we are looking at two angles that we need to trace in this 3D space to locate the side chain orientation. Because it's a 3D space, you need two unique angles to figure out what the orientation of the side chain is. So we record this pair of angles to describe the polypeptide backbone. That goes the description of the structure. And then is the description of the sequence. Now, if to connect sequence to structure, we need to encode sequence as well. So sequence is just sequence of alphabets. You cannot do math with alphabets. So you have to convert this alphabets to some numbers. So there's something called a transformer architecture, which can look at sequences and convert them into useful information. What basically happens is, let's say, if this is a valid sentence, let's say I am from India and he is from India, you can always understand that, like, based on what noun is used, it already forbids some combinations. You cannot write I is because you know the grammar, it comes across as wrong to you. So how about if I were to give you a book of several million protein sequences, which are validated protein sequences, and tell that each of them are sentences and each of these alphabets are the words that make up these valid sentences, And if you were to able to memorize all of them at some point, you would figure out, okay, there is this inherent grammar. These chunks or these alphabets should come after each other, and these are not allowed to come after each other. That gives us the contextual importance of why a certain amino acid is placed before or after the other. So we did some tests where we masked certain amino acids of given proteins and asked them to recover those masked amino acids, and we did that test. And for we looked at the NCBI conserved domain databases and dropped full domains from existing proteins and asked it to recover these full domains, trying to see if there are contexts that are set by the preceding and succeeding amino acids that actually determine the shape and structure and also the sequence of these domains. So once we did these two tests, we were sure that this model has something performative. So one thing that we do not usually say a lot is that this is also typically very fast, and it operates with 3% of the compute that AlphaFold works, while accuracy is not across the roof, but we are working towards it. So we are working at like several milliseconds per prediction at this point, which is very important for generating large libraries of structural proteins, and mean prediction times for AlphaFold you can see here. And there's one thing to be aware, it's regardless of any machine learning system, is that to probe the real biology that's there inside it. So one simple test that we did at that point with AlphaFold was that you generate a multiple sequence alignment that gives you this protein structure prediction. This is the structure for the top sequence that you look at here. The structure is over here. If you were to supply AlphaFold the flipped multiple sequence alignment, it will actually give you the flipped protein. Whether the flipped multiple sequence alignment has any sense or any meaning, that's up for debate, because if it's a very short protein, like the flipped sequence might actually be a completely new, fresh protein. And other cases, it would be an artifact, right? So a flipped protein and a flipped sequence does not necessarily mean anything. So we show that for cases where there is lack of information of related sequences, it's like orphan proteins for which if you do BLAST, you'll only get that sequence back, nothing else comes back. For those orphan proteins, we outperform AlphaFold on 58% of the sequences that we curated. And then there's another example, which is therapeutically relevant, I think leucine-rich repeat proteins. So if you use the basic AlphaFold algorithm, where you supply just the fraction of this sequence, not the full sequence, just the fraction of this sequence, it tends to recognize that, oh, this actually comes from this whole big sequence. It will give you the structure as if it's still belonging as a part of this whole big protein. So this can be somewhat non-physical, because what is it that's holding this little fragment in a way as if it would be held as is, as if it is a part of the bigger one. So one needs to take this with a pinch of salt. One needs to perform their right computations. One needs to relax this protein, perform molecular dynamic simulations, and do binding tests before you can confirm that the structure that you're directly getting right out of from the AlphaFold algorithm is right away correct. So having talked about the algorithm, I'll just finish off soon here, where we are saying that, we are currently looking at different types of pore engineering platforms for, membrane-based separations, drug delivery and stuff. One of the systems that we are interested in is the TMV mosaic virus channel protein, which has different several number of monomers, which are arranged in a spiral fashion. We are trying to optimize the number of these monomers and the different interactions they have to control the pore wall, selectivity, size, etc. And finally, I leave you with this thought that, now that you've seen this example of this, that I am from India, you should be able to figure out that if I tell you we have to add an extra word, which is not, based on your knowledge of grammar, again, without no extra information, you should be able to tell where does this not fit, and it's not an incorrect insertion. This is the correct one. So this is analogous to the thought process of doing a docking, like peptide protein docking or small molecule docking. There are multiple conformations which are incorrect, but there is one which is correct. So we are trying to use these kind of models to get at to the bottom of this. And finally, we just got started in fall 2022, but looks like we're starting to have some fun in 2023. I have a lab of currently 11, 12 people. So we have some students who are rotating and also collaborating from other labs. But this is where we are. Happy to take any questions.
Amazing talk. I love your explanation at the end of you just mentioned about the language plus versus the protein structure thing. That is very clear to me. So, Pete, you have any question, comment?
Well, first of all, outstanding talk. And, just to put this in context, his work in this Nature Biotech paper is just incredible. So it's really, first of all, just super exciting work.
Mm-hmm.
I am particularly interested in the last part in talking about the docking problem, just because that problem, you could argue, you and others are being so successful with these language models in predicting structures that it's not solved, but it's remarkable how much progress is made. Now, on the contrary, though, when we think about not solving the protein folding problem, let's say for if I think antibodies, so not solving for antibodies or whatever protein, but then the docking problem is such a significant one because it could be transformative, right, in the way things are done if you could solve that problem. And I've seen a lot less done there, particularly when we think about protein language models. And so I wonder if you could just comment on that, that where is the field, where are you, and what do you see as sort of the future and the challenges and opportunities?
Right. I think when we are talking about docking, so a couple of things. I know when you mention antibodies, you're thinking of the CDR3 loops, which are extremely flexible and they would be binding to different grooves and stuff. So we've had little exposure to CDR3 loops, and with our limited exposure, we've been able to see that if you take these loops as disembodied pieces of peptides, they behave very differently as compared to when you have the full variable fragment. So treating the CDR3 loop binding problem as a peptide docking problem is incorrect. So we need to get better with protein docking, first of all, and also identifying how small vibrations cascade down to different parts of the protein. So we are trying to do that, like on the molecular level. We are trying to figure out contact maps. The people were working with contact maps like 10 years back from now, but I think now we have better we are trying to get information theory kind of propagation of information down this nodes and edge based graph, where a node is an amino acid and edge is not necessarily the connected amino acid. It is the connected amino acid, but also something that's non-covalently linked. That would also constitute an edge. So, and that contact map would transmit not just the vibrational information, but would also transmit things like the electron cloud density based on the solvent that is there, the pH, solvent, and all of that. So we are we just getting started, but we are planning to look into that. We are doing that for much shorter proteins now, which are way more structured and easier to track, but that's definitely in the picture. One more comment on the language model side of things. So with language models, I gave a very simplistic, adding the not kind of a example. But there could be cases where if the sentence is big enough, you can have nots at multiple places, and yet it would mean the same right thing. So, and those would introduce false positives. You actually want to convey this thing, but you are able to not convey and not able to perceive whether you made a wrong answer, right? Wrong prediction. So those would be challenges to list, and figure out. But we could also filter those. If you have a wrong docked position, from active site chemistry that that's not where we are looking for. You can limit the search box. That could be also something, So there are ways around. We have some thoughts into it. We've only just gotten a lab together. We are trying to look into it more.
Yeah, maybe just to follow up on that. So, when you think of protein language models in the training sets that you have, which are massive sets of proteins, conceptually, it sort of makes sense that you could learn the natural context of some of these predictions. And I think you explained it very well. So if you take out, for example, a segment, and then you ask for the model to predict that segment, that it knows all these detailed context-dependent features. And so it gives you that. I just wonder, when you think about this docking problem, which again is a very hard problem, a very complex problem, What I'm wondering is that the training sets that you're using, presumably the complexes. So are there's different ways to think about this. There's learning from just protein sequences. Now, are there opportunities also to learn from natural complexes that would also be useful, or could you comment on that?
Yeah, my comment on that is that, so what we are currently doing over here is that we are training. You're totally right. We are first creating a very curated data set. We are not just trying to train on existing available data sets, because oftentimes they are biased, because let's say if you have more of alpha helical proteins, then you tend to learn more on them, overfit on them. You have more of beta barrels and stuff like that. So we're first normalizing on membership. How much of what we have is that like a fair representation. The second thing we are learning is that we can learn about complexes from single proteins itself, because at the binding interface, it's nothing different than a single protein folding onto itself, essentially. So you have two domains, like hot dog folded proteins like thioesterases and stuff. They have the beta fragments interacting with the alpha helical components. So if you just remove the extra chunk, if you are sort of able to delete it and study those interfaces, those are also stabilized interfaces. Those should give us way more opportunities to augment the data set, and then understand more about how proteins interact with each other.
And that makes a lot of sense. I mean, I think we're seeing reports now of people showing some of these protein language models predicting, say, mutations that increase a specific property, like, for example, in the antibody world, antibody affinity, and they do it, and you're without knowing anything about the antigen that you're targeting. So I suppose that's and so when you see that, they'll test, say, tens of mutants, and out of that, they'll find mutants that increase the affinity of the antibody. Now, in that case, though, they're not even considering the antigen. So presumably that's like an intrinsic, they're looking for mutations that sort of improve the intrinsic properties of the molecules. But what you're talking about is more sophisticated in the sense that theoretically, let's just say I asked you, could you predict mutations that would increase the affinity of an antibody, theoretically, setting aside some of the complexities of the loops? But any problem that you're tasked with a protein binding to another protein, let's say, you envision the way that could be used is sort of considering both binding partners, because you're saying you can learn from the interface that those binding partners would form. I mean, I think that's quite interesting.
Yeah, and also I think that's pretty much exactly, you summarized it well. I think what also happens is when you try to look at this language models, at some point they are connecting with a structural database also in their training process. So what happens is, let's say when it's learning from different antibodies, it's learning from that static PDB representation. So that PDB representation already has the memory of the conformation, which its antigen bound state. So that is coming as a conformational bias, which is a good bias if you want to predict antigen binding.
I see.
So the sequence already, it's not in its most native relaxed state that it's trying to predict.
Interesting.
Yeah, so AlphaFold actually has been shown, at least for homologous proteins, it's been able to show that even without the magnesium or zinc binding bound in the pockets, its pocket is somehow turning in a way as if it would accommodate a zinc or a thing.
Wow, fascinating. Fascinating.
Yeah, so
and that's sort of the bias you're saying of the prediction. I see.
If you're trying to design a magnesium binder, it's a good bias for you. But if you're trying to switch the from magnesium to something else, that's not a good bias.
Okay. I mean, the other thing I'm being told is that AlphaFold is not particularly good at predicting the impact of individual mutations on I suppose, something as simple as the structure, for example. I don't know, could you comment on this? And because this is an important issue. So routinely, we want to make variants of proteins with single or few mutations, and we want sort of accurate predictions of the structures. Could you comment on the challenges or the opportunities?
Yeah, absolutely, absolutely. I think so. What happens is, I know this. So we happen to be a part of a team who are also in the review committee of this paper, when it was being judged. So we had some priority. We were looking at this model up and close. So you generate the first step for these models is that you generate a multiple sequence alignment for the target that you're trying to predict. So moment you make a point mutation, your multiple sequence alignment does not change at all, because it's more of a noise than a signal. So it's the structure prediction remains exactly the same unless you've made drastic enough changes so as to be able to perceive it as, oh, I need a different sequence alignment for this. In which case you're talking a completely different problem. If you're making one or two mutations, even if you change the active site to a glycine, active site residue to a glycine, it would tell you that it has the same structure.
I see.
Right.
And are there ways to address that, or methods you're developing, or do you envision ways of because this is an important problem, frankly, that's kind of holding back the use of these approaches.
Right. I think I've not seen any method. So typically, anytime a new method comes out, that's the ablation test that I sort of force it through.
I see.
So what I've seen, the long story short, the idea is that if we ignore the last step where we can see the PDB up close and in visuals, if we ignore that step, if you look at the penultimate step before the PDB is popped out from the system, there is this mathematical representation which is which has a bijective map, which is a one-on-one mapping with the structure, but it itself is a huge matrix of numbers. Okay. That representation is much more information rich than the structure itself, because structure has obligations to remove clashes and stuff and to do things, and those often cascade down the backbone. So it's much more high fidelity in terms of prediction of effects of point mutations when we look at language models and look at their inherent latent representation, which is the mathematical representation before the structure is popped out. So we stop these methods from predicting the structure itself, look at the representation, and then do operations on that.
That's very interesting. And then you can interpret, there's information there you're saying about what the mutation could be doing that you can capture. I see. Very interesting. Okay, well, maybe I'll bug you about that. That sounds very interesting.
So cool. Wonderful. Do you have any other question, comment?
I mean, it's just super excited, and please, yeah.
Actually, I see some question from audience, so I will just actually my question already you answered, so I don't need to ask that question. So that's about how about the longer sequence, what happened, and that's already you answered. So Maya asked question about, do you have any opinion on using engineered protein pores for protein sequencing? That's interesting.
Yes. So protein sequencing, I think she's asking about peptide sequencing in terms of identifying the sequence of peptides. The greatest challenge, I think, is that, so there are peptide sequencing pores people are talking about. I think the efficiency issue is that when you're doing ATGC sequencing, you need to have 4 unique signals of current. When you're doing peptide sequencing, you need to have 20 unique signals of current to be able to describe the all possible amino acids distinctly in a peptide sequence. So that becomes a problem, because now you're asking questions about how do I distinguish the signal between a valine and a isoleucine, which are roughly the same size, and then same hydrophobicity, same everything, same sterics roughly. How do we have a thing that can do that? But I think right now we are not there yet. But in terms of peptide sequencing, one should be able to come up with sequence signatures that, like, okay, this part is hydrophobic, hydrophilic, aromatic, polar. These kind of broad classification based sequencing could be done, but I don't think there exists that can subtly separate valine from leucine or isoleucine.
Wonderful. So second question is, training set, is there interest in leveraging data set created by something like directed evolution or other highly multiplexed mutational studies? Do you have thought on whether the creation of certain data set would have outsized impact on prediction compared to leveraging the currently available data set?
Right. I think, I mean, everybody sort of has their own philosophy on data sets, but I think what we're trying to do in our lab is that, when we collaborate with someone, we tend to collaborate with experimental people for validating or, starting a study. Anytime any mutational data is there, we learn from the negative data sets as well. So if something does not work out, that is also useful information for the model to know that this is something one should avoid. If you're trying to engineer something for function, even if you're trying to engineer for stability, even if you're trying to engineer antibodies or something, you want to also incorporate not just the positive data, reinforce it with or penalize it with the negative data also, that you predicted this, but this does not work out. So that's one way of incorporating maximally whatever is done in the experimental side of things.
Wonderful. Pete, do you have any additional question or comment?
I'm just very excited and really look forward to following all this great work. And I'm impressed what a fast start you're making. And I really, if I can support you in any way in the future, please let me know.
Okay,
Thanks a
so
lot.
wonderful. Thank you. If I do not see any additional question from the audience, so I probably close because now it's 11:04, the Central US time. Okay, so thank all for joining and staying today. We'll meet again on April 20th, Thursday, the same time, the same Zoom link. We'll have Professor Yong-Soo Jin at UIUC and Dr. Corettin Bryot at ETH Zurich, the Switzerland, actually, which I will visit to give my talk next week. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speaker. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks, and I'm stop recording. Thank you. Just 1 second.